Hi, my name is Lindsey Giffey. I'm a junior from WF West High School, and I will now be switching into my PowerPoint explaining my project. My project is the effect of the duration of light regarding desynchronization on the period gene in Dorficilla melogastrum. Desynchronization occurs when people change time zones quickly through flying. This is commonly known as jet lag. Side effects are that it causes tiredness at unwanted times. The circadian rhythms of the person who traveled are out of sync with the new time zone that they changed into, which is what causes the desynchronization. Circadian rhythms are the body's natural clock that perform time-based functions. These functions are controlled by the period and timeless genes. The timeless gene creates a protein that regulates the body's circadian rhythms. It is light sensitive, so it is constantly being broken down during light hours. The period gene is the second gene that regulates circadian rhythms. For it to be stable, it must form a complex with the timeless protein. If no complex is formed, it is broken down by the double time protein. These proteins self-regulate the amount of their mRNA that is present in the cells. When enough complexes form at night, they move into the nucleus and form a supercomplex with the clock and cycle protein complex. This stops mRNA production. When light re-enters the cell, the timeless protein is broken down, the supercomplex is destroyed, and mRNA is produced again. Here's a diagram of self-regulation. It starts off with the mRNA in the cells. This produces proteins. They do not form complexes because of the light, so both proteins are being broken down. The light is then taken away, they form complexes, and move into the nucleus. Here, the mRNA forms a supercomplex and stops the production of their own mRNA. Light then re-enters the cell. The supercomplex is broken down, mRNA is produced again, and the cycle starts over. It was hypothesized that the group that was given light between 1 a.m. and 2:30 a.m. would be best adjusted because the cycle would be able to restart and adapt to the new light pattern if it was given light 1 hour before sunrise. I'm sorry, I accidentally jumped forward one too many slides. So this is my hypothesis page, and as I said, I thought that the group that was given light between 1 a.m. to 2:30 a.m. would be best adjusted. Here's the light variable groups that we have. We have a West Coast control and East Coast control, and different amounts of varying lights throughout the flight, as you can see. I separated the flights into 7 groups. 6 were adjusted to the West Coast time, and 1 was adjusted to East Coast time for 1 week. The flight was then simulated according to the groups on the previous page. Flies were then euthanized immediately after the flight. The RNaqueous 4 PCR kit from Life Technologies was then used to extract mRNA from the samples. To convert the mRNA to cDNA, TACMan reverse transcription reagents were used and was then ran on the PCR machine. Afterwards, 2 mastermixes were made for the real-time PCR reactions. One was made with the 18S control and one was made with the period gene custom assay. The reactions were then loaded and ran on the real-time PCR machine. Here's the amplification plot. The red, yellow, and green lines that are farther right are the period assay results, and the blue and purple lines are the 18S control and are closer to the left-hand side. This is because there is a larger abundance of the 18S mRNA in the cells of the fruit flies, so they are able to amplify faster. This is a chart that shows the average of when each group reached the CT line. These numbers were taken from the averages of the amplification plot. The mRNA extraction and reverse transcription processes were successful because after the steps were completed, their concentrations were checked on the nanodrop and there were good readings. This proved that there was mRNA or cDNA in the sample depending on the step being tested. The real-time PCR process was also successful because amplification occurred. All the samples reached the CT line before the 38th cycle, which showed there was moderate amounts of the period cDNA in the sample. The 18S sample reached the CT line before the 30th cycle, which proved there were large amounts of the 18S cDNA in the sample. This also proved the mRNA extraction went well. The mRNA levels of the controls did not appear as expected. The West Coast control should have had low amounts of period mRNA because it was in darkness during the flight. Instead, it had large amounts of mRNA in its cells. This caused them to have mRNA levels close to the East Coast time control. The samples also reached the CT line within 3 cycles of each other. Because this data is very close and the controls were not correct, no accurate conclusions can be made. If this project is redone, I would make the time change more drastic, such as a flight from the West Coast to Europe. This way the controls would differ greatly in the amounts of mRNA present and the data would be more spread out. This way, accurate conclusions could be made because the mRNA levels would not be as close. I would like to thank Henry Weeks, who is my teacher and was able to help me through my project. I would also like to thank Wendy Neal, who is Henry Weeks' sister. She was my substitute teacher when Henry was gone because of heart surgery. She also gave me great advice on my project. David Kogan from Life Technologies was able to give our class a discount on supplies, which was very helpful since our class was on a tight budget. I also like to thank my family for their support. Thank you for taking the time to review my project. I really appreciate it, and I'm excited to see you at BioExpo.